Today's lecture, The Chain of Command, is about the development of the nervous system. And one of the questions I want to look at is how much of your behavior is, as it were, hardwired, determined while you were an embryo, and how much of it is determined by your experience. Now, let's have a look and see what happens when you're born. We're going to see a film of childbirth. And the question is, how much do you come into the world with you? What can you really do at this stage? How much is really preformed and already set?
Girls, this is good. Another one.
Well, you see, you can certainly cry. And more than that, you probably know that if you offer a baby your finger, it can grip that finger, it can suck at its mother's breast, and so you already have some abilities already wired into the nervous system when you're born. Now, if Frankenstein is going to make an angel, he's got some real tough problems. He's got to design a brain, and here I have a human brain. He's got to design a brain that has an angelic disposition, if you believe an angelic disposition can be wired into a brain, or whether you think that only comes from experience. So he's got a big problem there. He's also got to get nerves to the muscles to move the wings in order that the angel can fly, and that's another big problem. And we'll at least have a shot at looking at some of those problems. Now, the brain is essentially made up of a very, very large number of cells. And there are probably more cells, if you just look at your thumb, there are more cells, if you took a piece of brain that size, took a piece from your brain that size, there are more cells in that than there are many more cells in that than there are people in the world. So the brain is really a combination of millions and millions and millions of cells. I, unfortunately, am losing the cells from my brain at about 100,000 a day. It's not very serious. I claim it makes no difference to me. There are others who may disagree. Those of you under 20 are in a more fortunate position. You haven't begun to lose your brain cells yet. And so if you're under 20, now's the time when you have the most brain cells you're ever going to have. So enjoy them while you have them. And part of the reason for this is why, if you lose brain cells and you don't replace them, is that there is no cell division of a nerve cell. Nerve cells just don't divide. And why the brain gets bigger from when you're a baby to when you're an adult is there's a little bit of cell division relating to the growth to more nerve cells, but it's mainly the nerve cells just getting bigger. Now, having a big brain in itself doesn't do you a great advantage. Now, it is true that Oliver Cromwell had a brain about twice this size. But there are many very, very clever people who have brains smaller than that. So brain size isn't important. The reason why it doesn't terribly matter, why size isn't so terribly important, is that the really important thing about the brain is connectedness. It's the connectedness between the nerve cells. It's connecting that really matters. And the cells, or many of the cells that you have seen and which you think about, have tended to be rather small, round objects. However, if we go along and have a look at some cells in the brain, and this comes from the eye region, you can see that they have very different shapes. And the feature, let's take this one here. Here's a cell, that's where the nucleus is, and you can see the processes branching, these extensions from the body of the nerve cell. Lots and lots of extensions. And they form a meshwork. And that connecting of the meshwork is the clue to understanding how your brain functions and how your brain is made. here are some details of some nerve cells, and just let's have a look at this one here. This is where the inputs come from other nerves. That's where the nucleus will sit with the genetic information, and then you have this long extension that goes over here and then branches really like a tree or like seaweed into many, 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 many different endings. And those many, many, many endings make connections with other nerves. And it's that complexity that is really important. We can have a look at some high power of these nerve cells. And once again, this is a scanning electron microscope picture. Here's the body of the nerve cell, and here are the long extensions that are going to go and make connections with other nerve cells. And we can see that in even higher power. Here are the nerve cell bodies, and look here, there's a nice extension going away. There are lots of them here. Here are other extensions going away to make connections elsewhere. So you see, you mustn't think of nerves as just small round bodies. You've got to think of them as a central part with extensions going quite long distances and making contacts with other cells. Quite a nice analogy for thinking about the nervous system and the way it's connected is to actually have a look at a computer, and let's have a look at the microprocessor. And if we look at that in higher magnification, we can begin to see some of the components. Take it out, and let's have a look at it. It's quite small. We can look at it as it's magnified. And you see all the components here are not all that different from some of those nerve networks that I've shown you. And if one can see here, if one looks at the activity in this chip of the microprocessor, if one could look in the brain, one might see very similar sort of activity. So it's connectedness and the interactions between the nerves that matter. When you think of nerves, I think most of you think of really rather large structures in your body. And it's perfectly true. If I prick myself like that, the impulse runs from there in a nerve all the way up here, round to the back of my spine, into my spinal cord and to my brain, and that's the way I know I've been pricked. Now, the structure that carries that message is very much like this cable. It's made up of many, many, many different nerve cells. And it's just like these individual ones here. If I tease out one, there, that would be a single nerve cell, that would be another, and yet another. So hundreds, even thousands of them, all together in one cable-like structure. And I think you can see this quite nicely here in this picture of a nerve cut in cross-section, and here you can see the individual nerves, the extensions from the nerves, and they're all bound together, and that gives you the nerve that you can actually see if you dissected out your arm. Now, this cable-like structure, this nerve, can be very long. Well, just let me make it clear to you. The cell body, which sends out those long extensions to the tip of my finger, that cell body sits up here. So it's a single cell, or lots of single cells, that send these very long extensions all the way down my arm to the tip of my finger. That's a couple of feet, that's not that much. Think of the elephant. With the tip of the elephant's trunk, there's a single nerve cell that has its ending at the tip of the elephant trunk that goes all the way up to the elephant's nose and into the brain. They really can be very long, and I think it's important that you realize that. Our problem then is to find out how the nerve cells find each other. How do they get connected? Now, in embryonic life, the nerves have a very special structure at their growing tip. And we're going to see that now. We're going to see it in individual nerves. Here's a growth cone of a single nerve in culture being speeded up in film, moving across like this. Another single nerve, here's the growth cone. It's splitting into two, but it's going to join together again, exploring the environment, trying to find, but they're not going to find it here in culture, their appropriate neighbors. Here's some more. There's another one there, another one there. These are all single nerve fibers. Their cell bodies are way up there with the nucleus, and their distal tip, this tip at the end, searching to make the right connections. And the problem is, how do they know how to make the right connections? How do they imagine them? And let me try and give you a feel of what it might be to be a nerve cell. Now, I have no doubt that almost all of you could pick out your mother from the 60 million people in Britain today. That's quite easy. It's an amazing feat. You could find your mother among 60 million people. You'd recognize her. Now, I don't say that nerves are that clever. They may not be that clever, but they certainly are pretty clever at finding out who they should make connections with. And in order to give you a feel at what it might be like to be a nerve, I want you to do the following. I want you to look at your neighbor, please, at their face. Okay, are you looking at your neighbor's face? Okay. Now, shut your eyes, please, and gently feel your neighbor's face and see if you can recognize. Gently, without putting out eyes, but gently stroking, just like a growth cone. Okay. Well, that's the problem that nerve cells have in trying to decide. They've got to recognize not their mothers, but they've got to recognize their neighbors or their future neighbors in order to decide where to go. Because what nerves don't use their eyes, they use the surface of those growth cones in order to decide. Let's have a look at how the early nervous system develops in the embryo. And it comes from a sheet of cells on the surface of the embryo which folds over to form a tube. And let's have a look at this in a frog. And here's it beginning to fold over, a sheet of cells. This is the bit that's going to be the brain, and that's going to be the spinal cord back there. And you see it folding over. There's the one edge and there's the other. This is the brain region, a bit broader and bigger. And there's the top bit of the spinal cord, the pieces coming together, the edges coming together to fuse to give you the spinal cord, and that's going to be the brain. And one can get a feel as to how that folding of the sheet occurs by having a look at this model here, in which you can imagine this is the sheet on the embryo, which is going to form the brain and spinal cord. And one of the ways in which it changes its shape is that there are on the surface, on one side of the sheet, there are little mini muscles, micro muscles. And if they contract, then because they're asymmetrically placed, the sheet will roll over and form a tube. And that's a very important process in development. And here you can see it here, you saw it there. And then, of course, those folds fuse together in the midline there, and then that's fine. Now, that doesn't always happen. And there is a condition that you get in children known as spina bifida, in which that process doesn't take place properly. And here you have an exposed bit of the spinal cord where that hasn't closed over properly. And fortunately, surgeons now are extremely clever and ingenious in being able to correct that abnormality. It's also true that research has shown that one of the ways of preventing spina bifida and other malformations in the closing of the neural tube is for mothers to take vitamin supplements. And that rather simple treatment has really been a major advance in preventing those sort of abnormalities. We've seen that there's a sheet of cells that folds up on the surface of the embryo. But why that sheet of cells? Why is that the group of cells that's going to form your brain? And what's more, how does it become integrated with the rest of the body? Just think of the problem. The nervous system comes from a sheet at the top, but all your bones and your muscles and your heart and everything else comes from material inside. How do the two integrate? How do they become coordinated? And the answer to this comes from work that was done something like 50, 60 years ago by a very famous German embryologist, Hans Spemann, who showed that it was because other tissues came to lie beneath the developing nervous system and ordered it to become a nervous system that it became a nervous system. You can see here the tissues moving in. This is again a frog embryo. The nervous system is going to come from the other side, and cells are going to move in from this side, move into the embryo. Here they're beginning to move in. You'll see it quite nicely. Just moves back in a moment. Here the cells are moving in, and they come to underlie the future nervous system, come to lie underneath it, and then they order the tissue over them to form a nervous system. And those cells that move in, those are the ones that are going to give rise to your vertebrae and your muscles and really the main structures in your body. And that, of course, makes sure that the two actually are in the right position. And people were very excited about that when they realized that. And then they did an experiment. They said, well, look, if this tissue can order this tissue above it to become a nervous system, what will happen if we graft in a second piece of this tissue? What's going to happen? And let's have a look at that experiment. So here's a normal tadpole with its nervous system, its brain here, and its spinal cord running all the way down there. Then one does the experiment of grafting in an extra piece. And here we get an extra nervous system, another brain, and another spinal cord. And so that was terribly exciting. Here you can just graft in another piece of tissue, and you can get an extra brain. If only they felt they could find out what that substance was, what is the nature of the order, the chemical nature of the order, they would really be at the heart of understanding how to make a brain. So one of the first things they did is they killed the tissue to see if it would still work when it was dead. And they grafted in dead tissue, and they were thrilled to discover, yes, it still worked. Didn't make a whole brain, but it certainly made nervous tissue. Then they tried other things. They tried the liver of rat, and that worked too. Then they tried various chemicals from the bench, and that worked too. And to their horror, everything began to work. And then someone put in a small bit of chalk, and that worked too. So clearly there was something of a problem of finding this magic substance. And the reason there was a problem is that what this is doing is it's giving an order, but it's not giving a recipe. It's ordering from a menu. This tissue here is poised either to be skin or to be nervous system, and it just needs a little kick to be made to be nervous system. So in other words, the order can be really rather non-specific. And I must confess that even today, we still don't know the chemical nature of that signal. The signal goes to that tissue, rather a non-specific one, we think, decides to become nervous tissue, and it begins to fold over and it forms a tube. So we've now got a tube of cells that are going to give rise to your brain. The cells divide. At this stage, the cells are capable of division, and some of them develop into nerve cells. These nerve cells then put out their extensions, and they move to different parts. They explore, they move over some of the supporting cells, they climb up cables, they cross over other cells, and do that feeling that you were actually experiencing in order to try and find out where best they should end up. And they form, in most parts of the brain, a very well-ordered, layered structure. And here I've got an example of it from one part of the brain, the retina, and you can see that it forms a very beautiful layered structure. And let me show you what happens. Over here, you have the cells that receive the light receiving cells, and these are extremely sensitive. The eye can pick up the smallest possible quantity of light that you can think of. One photon can activate a cell here, which can then send a message down this nerve, for example, here, and then there's a whole other layer here with interactions between the cells, so they begin to process the message, which then passes on to another group of cells in this layer here, which then passes into this rather dense, mossy-like layer here, cells, quite a lot of the information in the eye being processed, then down into another layer here, and then finally out here into the nerve that actually takes the impulses to the brain. Quite a complicated structure, and they're highly layered. And that's true of almost all parts of your brain, even more complicated. And the point is that if something goes wrong when those cells are moving to their different positions, then you can get abnormalities. And here I have some mice which actually show abnormalities which are due to the fact that the cells really haven't gone to their right position. That in that feeling process and in looking to see where they should go, some of them have gone to the wrong place. And this first lot here are called shakers. It's a mutant. It's a single gene. And I'll take the top off, but I'm nervous they're going to jump out because they're very shaky mice. Can you see them shaking? They're very active, and they tend to have this almost not like a tremor, but they certainly shake. And you can see that one doing it there quite nicely. So that's one effect of the cells not taking up their right place. And let me show you another one. These are lurchers, and they are much calmer, but they have a little abnormality of movement. Let me see if I can get one out. Let me put my glasses on. Come. And I'll put you there. There we are. And if you look carefully, you see they have a slightly strange movement. There is that tendency to walk backwards. I don't want you to go off the desk. And they slightly wobble when they move. And that's the result. That's the sort of thing that can go wrong. Once again, just due to a single gene being somewhere faulty, leads by a long sequence of events to the nerve cells being in the wrong place, and therefore you get a behavioral abnormality. What I want to do then is to look a little bit more detail as to how the nerves in fact do find their way to different parts of the body. And in order to do that, we'll have a look at the spinal cord and how it connects up with the muscles. Here we have exposed is the back of a model, and you can see the spinal cord running all the way down here. And you can see the nerves from the spinal cord coming off in between the vertebrae here. If I look carefully, there are some more, and if I go a little further down, you can see them, perhaps slightly better. You can see them coming off there, and there, and there, and there. So nerves are coming off, and particularly down here, I want to look at the arm. You can see nerves coming off from between the vertebrae and going down to the arm. And we want to understand how those nerves, and let's look at those that actually go to muscles, how they find the correct muscles. And in fact, if they do find the correct muscles, and how clever they are. We have a look at this in the chick embryo. And just as I showed you there on the human model, okay, here we have an embryonic chick embryo. There is the embryonic chick wing bud. That's going to give rise to the wing of the chick. Here's the spinal cord, and here are some nerves leaving the spinal cord. They've been specially stained. They've passed through this region here, and here you can begin to see them at an early stage moving into the limb. And if we have a look at the tip there, you can see almost the growth cone as they're exploring what bit to connect up with. This one's going to go towards the end of the hand, and that one's going to go towards the end of the hand. And they have to correct up to the right muscles and the right part of the skin. And here are some others, just slowly at this stage, moving in. Now we can do an experiment. We can try and ask, how clever are these nerve cells at finding their way? What will happen if we put a barrier in at this point here? And let's have a look what actually happens. The nerve comes along, bumps into the barrier, says, oh, well, too bad, let me try and find my way around, moves up here, finds a way over the wall, crosses over, and continues along its normal course. And we can see that in higher power. Here it is. There's the barrier. And over here is the nerve. It comes along to the nerve, can't get past, moves to the side until it can find its way over, and then continues, branches into the correct place, and moves towards the tip of the limb, and so makes things perfectly normal. So nerves are really quite clever, not always totally clever, but nerves are really quite clever in being able to find the right path. One of the peculiarities of that system that I've just been speaking about is that you might think that the nerves move in, make connections with the right muscles, and everything's okay. But that's not the case. Because within a few days, about half of the nerve cells that have moved into the limb drop dead. They simply die. And the question is, why do they die? And one of the reasons people thought that they died is that they thought of them rather like travelers in the desert, longing for water and food, coming to an oasis, but the oasis not having enough goodies or food or nutriment for all of them, but only for some of them. So there was competition for these goodies, and those that didn't get it dropped dead. Those that got it stayed on to become fully fledged nerves. And the problem was then to find out what the nature of that chemical was which enabled the nerves to grow and to live. And in order to tell you how this was discovered, because the substance now is known, at least one of the substances is known, I want to tell you a story that actually has got 3 characters in it. If I could look at the first character, and here she is, it's this lady here, Rita Levi-Montalcini, who this year got the Nobel Prize in Medicine, and she got it with another person, Stanley Cohen, seeing here receiving his Nobel Prize. The two of them got the Nobel Prize for medicine for discovering a substance known as nerve growth factor. But as I told you, there's a third character. And let's have a look at the third character. And the third character is a snake. And David Ball from the zoo has very kindly agreed to bring his snake, or the zoo snake, along to join us. And I'll now explain to you how these 3 characters came to get the Nobel Prize. No, they didn't give one to the snake. The story goes like this. They wanted to understand what was the chemical that enabled nerves to survive. And they had noticed an earlier experiment in which a mouse tumor had been grafted into a chick embryo, and this mouse tumor had caused nerve cells to grow. That was surprising. They thought, gosh, maybe there's some substance in the mouse tumor that causes cells to grow. All we have to do is to isolate it, and we'll have our substance. Now, it's very difficult to do that experiment on the embryo, so they developed a very clever test system. And what it involves is taking a small bit of nerve tissue from the chick embryo, putting it in culture. There it is, and it just sits there in a rather boring sort of way. But if you put the right substance from that mouse tumor on, then the nerves grow like mad, and you get this proliferation, this large number of extensions. So you've got a very nice assay now for finding out when you do have your substance. So there they were trying to purify the substance from the mouse tumor. Now, in one stage of trying to purify it, it was necessary to destroy what they thought was one of the chemicals that was interfering. And to do this, they went along to the snake. Now, the reason that they went along to the snake is that snakes have in their glands, or poisonous snakes, venom. And venom contains chemicals or substances that break down other chemicals. And that's what they actually wanted. So they were using snake venom to purify their particular extract. Now, when you do an experiment like that, you've always got to do the control. In other words, you must try the snake venom without the extract. And to their amazement, when they took snake venom and put it onto their cultures, the nerve cells loved it. They grew like mad. So they were very pleased that the snake, they now concentrated on snake venom. But that's a little tricky working with snakes. And one of them had a very bright idea. They said, look, what is nerve growth factor doing in snake venom? It must be in other animals too, surely. It doesn't make any sense to have nerve growth factor in snake venom. So they said, maybe it's present in glands that are similar to those in the head of the snake. So they went along to the mouse and have a look at the salivary glands of mice. And lo and behold, in the male, not in the female, in the male salivary glands were packed full of nerve growth factor. And that, of course, made it terribly easy for them to isolate the substance, which is a protein which acts at extremely low concentrations, something like 1 molecule per cell. If that's one of the magic substances that enables nerves to survive. So the snake was an enormous help in that rather strange story of how they got nerve growth factor, and they well deserve the Nobel Prize. Thank you very much for bringing us this beautiful snake who rattled away so very nicely.
Not at all.
Very grateful. Thank you very much. Nerve growth factor's fine for survival, but it still doesn't tell us an awful lot about pathfinding and feeling. And to do that, to look at that a little further, I want to look at the eye and its connection with the brain. And this is a cross-section through the early brain. There's the original tube which I told you about. And your eyes are outgrowths of the brain. Here's the one side, an outgrowth of the tube here, and here's the other side, an outgrowth there. It grows out, and in fact the lens is going to form here because this orders this tissue here that's going to be the retina, orders this to become a lens. And it begins to develop into a lens. This is now going to be the retina, and it goes on developing. Here's the retina further developing. There's the lens that's been ordered by the retina. And we're now going to get nerve fibers growing back from here to make connections with the brain, and we see that here. This is a later stage. The lens is now formed. And here from the retina, the cell bodies sit in here and send out their very long extensions quite a long way back to make the right connections in the brain. And I think you can see what these connections look like and over quite what a distance they eventually come to rest if we have a look at this model here. And I think you can see very nicely that the nerves from this eye travel across the brain right across to this side here, to that part of the brain, and also give connections to this part of the brain. And this eye here gives connections not only to this part of the brain, but to the opposite side of the brain as well. So there is a very well-defined pattern of connections, and I've just tilted forward so that you may be able to see it even more clearly. Quite long connections between the eye and the various parts of the brain. Now, that's very important for the way you see, and we have someone here whose connections aren't quite right, and that has quite important implications. And the person, or not the person, but the animal whose connections are not quite right is Esme, a Siamese cat who Mrs. Gregory has very kindly brought here. And if we look we'll try and get Esme to look at us in the right way. Esme, are you going to look at me? There we are. That's very nice. Esme, you're looking very nicely. Let's try again. That's really very nice. Thank you very much indeed, Esme. And why that beautiful cat Esme has such a squint is because the connections between the eye and the brain aren't quite right, and it tries to correct for them. When we as people have squints, that's a muscular problem and has nothing to do with the connections between the eye and the brain. Let's pursue the problem of the eye and the brain, and let me show you this newt. Now, I have no idea how hungry it is, but let's see what happens. We've got a newt in here, and are you hungry at all? Yes? It's lifted. Ah. Come on, you're going to have to work for it. Come on, up you come. You want it, you're going to swim for it. Okay, and what did you see there? I think very clearly that when the newt saw the bit of meat above there, it lifted its head. And the newt is born with that ability. And what we want to look at now is some experiments related to that and related to the connections between the eye and the brain so that the animal behaves in a very sensible manner. Here is a model of the eye of the newt and its brain. And there are connections which run, the nerves run from the eye quite a long distances and connect up to the brain. What we can see here is that if we follow these red ones, they get tangled up in here, but nevertheless end up quite nicely in this part of the brain here. On the other hand, if we look at the blue ones, they run here and end up in this part of the brain here. Okay. Let's have a look now at what happens when the newt sees the food, for example, of the kind that I offered it. If I show it a fly like that, yes, where does the image fall? The image goes through the lens to the bottom here, to the bottom, this side here, it goes through the lens to the bottom there, and let's see what happens then. It's on this side, it's the bottom end of the retina, the bottom of the retina that's going to be activated. It's the blue ones. The blue ones activate these cells in the brain up here. It thinks it sees a fly up there, and it lifts its head in a nice and respectable manner in order to get the food. Okay. And we've got a little film here. Actually, our newt did it very nicely, but just look again. This one does it rather better. How, when it sees the prey up there, it lifts its head, and that's built into its behavior. It doesn't have to learn to do that. That's built in from the moment that it's born. Okay, so that's normal behavior. What we want to look at now is what happens when we do an experiment, and now I need a volunteer, please. And I just need to see. Would you come? Thank you. If you come and stand around this side, and I'll tell you what you've got to do. The experiment is the following. Here we've got this thing. What we're going to do is to do two things. We're going to cut the nerve, in other words, cut it in half. Not in half, we're going to cut it here, and then we're going to rotate the eye. So could we cut the nerve by you unplugging all of those, please, for me? Just do as fast as you can. That's all right, don't worry. What happens when you cut the nerve like this here is the cell bodies are in this part of the eye, and they survive perfectly happily. All those over here, now that they're cut, I'm afraid are going to drop dead. All right, there's no hurry. Terrific, one more. Lovely, they're all dead. Would you unplug them? And just hold that when you take them out, that's all. We don't want it falling over, that's all. Thank you. So the question is, what's going to happen now to this animal? Well, the first thing I can tell you is that these are going to grow back. The question is, how are they going to grow back, and where are they going to go to, and how's the newt going to behave? After that is, I have rotated the eye. So I've rotated the eye like that. The eye is rotated. All dead? Oh, that one's got a bit stuck. That's all right. We can just put them down there. We don't need them. Okay? Dead. Nice, healthy nerve fibers, rotated eye. Let's have a look now at what the newt does. The newt recovers. This is an experiment done, about 20 or 30 years ago in America. Have a look at this newt now. Offered the prey, moves its head in the opposite direction. So when you offer it a bit of liver up there, it goes downwards. It's going to remain a very hungry newt because it never really learns to correct. And let's try and understand why it doesn't, because I want you to reconnect. Will you do the blue ones first? And in fact, what happens is the fibers go back to their original position. They can find their way through the maze of things that are in fact here to their old position. Will you reconnect? You could just pull them out. I'll hold the ball. Yeah, so you could just pull them. Lovely. Terrific. Oh, and then you, fine. Be careful, you just have to hold the board. Lovely. Terrific. Growth cones at their tips. What he's plugging in is the growth cones. that we saw. Okay, and the red ones too, please, they go back to theirs. Lovely. And so, in spite of all the entanglement here, the cells can feel their way back to their original position. Okay. Well, we might as well have a yellow one as well. Don't have to do it. Just one to show. And let's have one green, just to show. Like that. And so what we've got back now is the cells have gone back to their original position. And just as before, there's a one-to-one correspondence between the image on the retina over here and the activation over here. And now let's have a look what happens when we show it a fly. Once again, here's this hungry newt, and we show it the fly up here. The image goes through the lens and goes to the bottom of the retina here, because that's the way it will go through the lens. It goes to the red ones. Let's follow the red ones through. It goes to here. It thinks the fly is down there now, puts its heads down, and the fly escapes. Very grateful. Thank you very much indeed for your help. The newt never learns, never corrects, because that's built in, and those structures remain like that forever. What we thought we'd do now is to do a little experiment that involves having tea and cake. Thank you very much indeed. Okay, let's put the cake here, and I now need a volunteer. I wouldn't volunteer that quickly if I were you. I'm sorry. I'm sorry, I'm just... In the spotted, yes, please. Keep that for the cake, put the knife there. Sorry, what's your name?
Joanna.
Joanna, thank you very much for joining us. Joanna, all tea parties are not what they seem, I should warn you. Would you mind putting that on? There we are. Thanks very much. Okay. And what I'm going to ask Joanna to do, if she's willing to do so, is I'm going to ask her to put on these rather special glasses. I'm going to turn you into a newt. Because what these glasses do, if you don't mind doing it, is they're going to invert. Everything you see will be seen upside down. And then I'm going to ask you to pour yourself a cup of tea. Do you mind doing that? Can you try them on? Just let's see, because they're quite heavy. Oops, I don't want to get... I'll leave you to put them on. Have you got them on? No, you've got to put them over your nose, probably. There we are. How's that? And you've got to hold them all the time with that hand. Do you feel all right? It's a little odd. You okay?
Yeah.
Sure? Okay. Now, what Joanna sees when she looks through there is that she sees everything upside down. And what I'm going to ask her to do now is, Joanna, do you think you could try? Don't fall over. Do you feel all right?
Yeah.
Do you have a try in pouring me a cup of tea? Let's see how you do. Don't worry. It's not hot tea. Don't worry. I mean, we wouldn't let you do that. You just go, gosh, you're doing terribly well. See what you can do. Here's the cup over here. There we are. Doing very well indeed. Try any cup you like. I don't want to make it difficult. Yes, that's okay too. Any way you like. Go ahead, yes. Can you tip it up? Go on, pour. It's jolly difficult. Go ahead. Just pour anywhere. That's... Well, rather milky tea. Feel all right?
Yeah.
Hold on, Joanna. We'll give you one more go. Hold on a sec. You did extremely well with that. But because you've been so brave, and you didn't break anything at all, as you can see. But what I'm going to do is offer you a piece of cake as a reward. So I'll quickly cut the cake. I won't let you do this. Could you put them on again? Can you manage to get them on? All right? Can you see me cutting it?
Yes.
Okay. I'll hold it up for you, make it easier. There we are. Thank you very much. Take the cake's yours. She certainly deserves the cake. Thank you very much indeed, Joanne. Now, incidentally, if we kept the glasses on Joanna long enough, within a week she could ride a bicycle. In fact, it's quite easily, unlike the newt, you can learn to do it. The trouble is, of course, when you take the glasses off, you've got to learn to do it the other way around again. So the nervous system, our nervous system, unlike the newt's, is really quite highly modifiable. I've told you that the newt can't do it because things are, as it were, hardwired in the newt. It's not that the newt can't learn anything. It can learn all sorts of things, but there are certain movements it can't learn. And one of the questions we have to ask is how much of our behavior built into us from the moment that we're born? And let's have a look here at a peregrine falcon about 10 weeks old learning to fly. Doesn't learn to fly, of course. Here's the watchful mother, rather anxious about its young who's been flapping its wings for some time, but has never, there's the mother watching it, this bird has never flown before. A few practice bouts, looks down the cliff, which it's somewhat anxious. I'd be anxious if I looked down and saw where it's going to fly from. It's a long way down, and off it goes. Mother's very happy. Birds can fly without actually having to be taught to do so. To what extent is that true of other animals, particularly the sexual behavior of rats? Here you have two rats, male and a female, and you can see that their behavior of the female and the male is completely different. And the important thing about this, that's there from birth, and that is built into them. And the reason why the male behaves differently from the female is due to the fact that the male rat's brain has been exposed to a hormone known as testosterone. Effectively, all of us have female brains, and those of us who are males, part of our male behavior, I would suggest to you, is due to the fact that we have a Y chromosome, therefore we have a testis, which therefore results in the production of this hormone testosterone, which changes, in the case at least of rats, the behavior. Because if you take a newborn female rat, inject it with testosterone, it does in fact behave completely like a male rat. So Frankenstein really does have quite a lot of problems. But for example, if he's going to try and change the genes in order to make some sort of difference in this structure here, he's going to have to deal with a very large number of different cellular interactions. And how is he going to arrange that each one of these tiny little connections between the nerve cells is right? And what's more, how is he ever going to know what effect that's going to be on our behavior? And let me try and explain to you the problem with an illustration from a game known as the life game. The problem is this. Can Frankenstein, who wants to construct something, can he ever predict by changing one of the genes at the beginning of the chain in development so that he can actually work out what the behavior of the animal is going to be? Now, what I'm going to suggest to you that even with very simple games or with very simple interactions, like something called the life game, there's no way in which you can do this. Let's take a game with some very simple rules. If a piece is like this, it's too lonely and it dies. If a piece has got 3 neighbors, it lives. That one will live. If it's got too many neighbors, it dies of overcrowdedness. Very simple set of rules. And what we have here is we've set this game up on a microcomputers, on BBC Micros, in order to see how, in fact, that's going to behave. And Paul, could you just show us what you've got set up here? It's called the life game. could you actually set the pieces like that for us, please? Anything you like. And then we'll start it with those very simple rules. Run it. Let's see what it looks like. Stuck. Let's try the next one. I mean, not that it wasn't interesting. It just ended up there. Matthew, let's see yours. Oh, you've got few of them going. Going very nicely. Are they going to die? All gone. Death is the result. Thank you, Matthew. Elaine, can we see yours? Oh, you're starting with quite a complex set. And they're doing interesting things. Rather lovely patterns. And it's got surprising results. The point that I'm making, there's no way which you can predict from the starting condition what these are going to look like. The only way you can find out is to simulate the whole thing on a computer. And how's Frankenstein going to simulate every bit of development of the nervous system on a computer? This one is, well, some of the bits up the top are stuck. Ah, we've got there. Very nice pattern we had. Thank you very much indeed. And what we've got over here is a new kind of machine, which David Alden's going to explain to us.
Well, as you said, this is a new kind of machine which we call the computing surface because it, rather like the brain, consists of an arbitrary number of transputer microprocessors, thousands if you like, which are connected together in many and complex ways, like the nerve cells you've described earlier. And this enables us to focus all these processes simultaneously on tackling any particular task.
And what we've asked David to do is to program a very simple equation. X squared plus y in relation to a Mandelbrot set. Don't let that worry you, but a simple equation, x squared plus y, machine works that out and substitutes it for y and does it again and again. You couldn't do it on the BBC Micros. You could, but it would take far too long because I think your machine does it so much faster.
About 3,000 times more powerful, yes.
And correct. How many calculations?
Well, to produce a picture rather like the one here takes about 1,000 million calculations. And if I change the color values, which are the result of that equation, you can see how really remarkably complex and quite beautiful this very simple formula leads us to. And in fact, if we were to magnify up any particular area of this 100-fold, you'd find as much complexity again in there revealed, but only when you can obviously do thousands of millions of calculations in a reasonable time.
Thank you. The point that one's trying to make here are two things. First of all, that very simple rules can generate very complex patterns. But from Frankenstein's point of view, the real problem is you can't predict what the patterns are going to be without actually simulating the total system. And that leads to real problems. And we'll have a look at some of his other problems tomorrow when I talk about growing up and growing old. Thank you.